I'm April Pyle. I'm an associate professor in the Department of Microbiology, Immunology, and Molecular Genetics, and also a member of the Broad Stem Cell Center here at UCLA and the Center for Duchenne Muscular Dystrophy. And I'm really delighted to be here today with you to tell you about one of my passions in the world, which is stem cell biology. And really, one of the focuses of my work in general is thinking about how we can harness the potential of stem cell biology for regenerative medicine. And this is really a new era of personalized medicine. And what I think that you're going to learn at the end of this is how relevant this is to muscle biology and to muscle diseases, including Duchenne muscular dystrophy. So I'm going to start out really by telling you properties of stem cells. So what is it that makes a stem cell unique? And really, where do these stem cells come from? So you've heard a lot about stem cells in the popular press, but what you may not really understand is that each stem cell might have a unique property, might have different properties depending on the source of these cells, and definitely the stem cells that are affected in diseases, including muscle diseases, are going to be different than how the stem cells behave in other diseases. And so those are key concepts that we're going to keep coming back to. But first, let's step back. Let's think about you're a stem cell biologist, you're an aspiring stem cell biologist, and you want to instruct others about the importance of stem cell biology. So you've been approached to think about writing for the Stem Cell Chronicle to really describe what stem cells are. And so what I want you to do is to take a minute and think about these questions and submit these to a contest to ask, really, are all stem cells the same? This is a really important question. What makes stem cells different? Are they all the same, or are there different properties that make these unique? And then the second question or topic really is, what regulates stem cell fate? What gives these cells the ability to behave differently in culture? And later we'll talk about their properties in regenerative medicine. So these are some questions that you want to think about, and so take a few minutes to write some answers to these, and then we'll come back to that. So let's just jump in to think about different properties of stem cells. And one of the most important properties is that a stem cell is just a cell that has the ability to make another copy of itself, so to make an exact replicate. This property is called self-renewal. So only stem cells are endowed with this property, which is remarkable. It means that these cells can expand, proliferate, and indefinitely give rise to more stem cells. So it's a single cell that can replicate itself. And in addition, not only the ability to copy itself, but also to specify, to make functional cell types. So what is a functional cell type? A functional cell type is something that carries out all of the important, the important workhorse machines of the cells, neurons, blood stem cells, liver cells, all of the unique properties that that organ systems have, but stem cells have the ability to make these functionalized cells. Now we'll talk about different properties of stem cells. Not all stem cells make all cell types, but in general these are properties that you need to be aware of, which is that they can self-renew and differentiate. So let's think a little bit about different types of stem cells, their origins, how they are unique or different, whether or not they're embryonic or adult. So these are the two main types of stem cells. Their origins, they're either an embryonic origin, an embryonic stem cell, or they're an adult stem cell. Now, embryonic stem cells are unique in that they can differentiate. They are endowed with the ability to differentiate into any cell type in the body, and this is termed pluripotency. In contrast to adult, where typically these cells only are endowed with the ability to make a specific cell type within that organ system. So we'll talk a lot today about muscle stem cells. These are adult stem cells that have the ability to make cells within the muscle. Embryonic, however, can make any cell type in the body. And we'll also talk about how those can be harnessed for thinking about muscle diseases. Now, where are these derived from? This is important. Embryonic stem cells are really an in vitro artifact. They're derived from the inner cell mass of the blastocyst, and this is in contrast to adult stem cells where these are derived from tissue-specific sites, right? So muscle stem cells would be obtained from the muscle. Neuronal stem cells would be obtained from the brain. So they're obtained from different sources, and their properties are different, and the numbers of stem cells are different. Because embryonic stem cells are an in vitro artifact, they can be cultured indefinitely, whereas adult stem cells have a limited potential. They have only a specific or a finite division time, right? So those are key differences. Embryonic or immortal adult have limited expansion potential. So there's pros and cons for thinking about that in the context of using these in regenerative applications. Now, what about getting these cells to differentiate? Well, the best way we test this is to see, if they're pluripotent, which means they can differentiate into all three germ layers, endoderm, ectoderm, mesoderm. We ask this by testing the ability of these cells to do this in vivo. And embryonic stem cells have the ability to differentiate into all three germ layers, really any functional cell type in the body, by formation of what's called a benign tumor or a teratoma. Now, what you can do is isolate these cells and look and see how many cell types have these embryonic or pluripotent stem cells differentiated into. Now, you'll see all cell types essentially in these tumors, but in adult, if you were to put these cells in the same assay, they would not form cells from all three germ layers. They would maybe only form specialized blood cell types if they're a blood stem cell, or neurons if they're a neural stem cell. So they have differences in their ability to form tumors or teratomas. Another important difference is that, a lot of what we know about using stem cells in regenerative medicine comes from the wealth of information in adult stem cells, particularly the hematopoietic stem cell system. And we know how to control their ability to differentiate into these specialized cell types, into individual blood cell types. So blood cells can differentiate really well into blood-specific lineage cells, B cells, T cells, macrophages, right? And that's cell type dependent, so they only differentiate within that lineage. However, embryonic, as I told you, can differentiate into any lineage, right? But how do you instruct an embryonic stem cell to make just that lineage and not the other lineages, right? So that's a key question that we still don't understand, that's poorly understood, and really is the focus of a lot of research. And so we're really at an exciting time now where we understand differences, we know a lot about the properties of these different stem cells, and we really want to think about how do we move these forward in disease applications, in regenerative medicine, in disease modeling. And what people are doing now are thinking about differentiating specialized cell types for clinical use. Adult stem cells have been used routinely in clinical applications, specifically for blood stem cell applications. But embryonic, this is less well understood because, as I just said, it's not clear how to differentiate these into specialized lineages. So we still have a lot of work before we're thinking about using these, and again, depending on the disease setting. So what I want to do now is really focus on specific sources that might be relevant to muscle diseases for Duchenne muscular dystrophy. Now, as I told you, adult stem cells have a limited potential to differentiate into those specific cell types. There are many different adult stem cell sources. The most well understood is the satellite cell. This is the endogenous muscle stem cell that we'll talk more about. This cell will differentiate into another progenitor cell, the myoblast cell. These are derived from muscle themselves, so they're going to be happy in the muscle environment. Other types of cells that have differentiation potential include mesangioblast stem cells that are derived from blood vessels, mesenchymal stem cells, which can come from many different sources, stromal, bone marrow, fat. So depending on the source of where you isolate these stem cells from, this might give the cells a different property. And we'll come back to how that source regulates the stem cell itself, because this is an important facet of stem cell biology. There are also CD133 positive cells, termed so because this is how they're isolated and characterized, and they have different origins, either in blood or muscle. Another really exciting type of stem cell is a pericyte cell, which actually, has the potential to be important in a lot of different regenerative aspects, and they're also derived from either blood or muscle. Now, a lot of research has been focused on many subpopulations within muscle, muscle-derived, or what are called SP cells, side population cells, and all of these are different types of potential adult stem cells that could have relevance in the context of muscle disease. So let's focus for a minute on the endogenous stem cell of muscle, the stem cell that has been the most well characterized for skeletal muscle, the satellite cell. The satellite cell has really well characterized markers that we use to identify the endogenous stem cell. Another important property is the location of the satellite cell in muscle. So the satellite cell expresses a key in the adult, a key transcription factor called PAX7, as well as other extracellular surface markers that we'll come back to that we can be used to identify these cells. And importantly, the satellite cells have a really unique location. They're located between the sarcolemma and the extracellular matrix of muscle. And this location, as we'll talk about, endows them with unique properties to really become activated, which means that they've obtained cues or signals to tell these cells, hey, it's my turn now to get in there and regenerate in muscle. And we'll talk about the steps and how they do that. Once they get those cues, those signals, they leave a quiescent state, they become activated, they turn on another transcription factor, MyoD, and this is a real driver of muscle differentiation and is well studied. And then they have the ability to differentiate to make more muscle. But its properties start from the original stem cell and muscle, the endogenous muscle satellite cell. So this property of the satellite cell to really activate upon signals, upon injury, allows this stem cell to differentiate into another cell, the myoblast cell, which eventually will give rise to differentiated muscle. Another unique property of the endogenous muscle stem cells, or the satellite cells, is that there's a lot of interesting biology still that we're learning and understanding about these cells, which is that, again, their location is interesting because they can often be seen near the motor end plate in isolated myofibers. And so what this tells you is that these are closely talking to other signals at the neuromuscular junction, which you can see with the arrowhead next to the satellite cells shown in green or with PAX7 staining. And so I hope you can appreciate that they have a unique location, and these stem cells are endowed with unique properties, specifically in the context of skeletal muscle. Now, satellite cells, in addition to their unique location, have a really well-defined, in fact, the satellite cells are one of the most well-characterized adult stem cells because we know a lot about, in the adult, how these cells transition from a quiescent satellite cell to a terminally differentiated myofiber. And this has been done by a series of experiments really testing what are the important factors, transcription factors in particular, that are required to regulate stem cell biology or the satellite cell. For example, if you target some of these transcription factors, PAX7 or MYF5, you'll see that over time the satellite cell behavior either is changed or the satellite cells are lost over time in development. And so, we know a lot about these transcription factors through the process of differentiation. PAX7 turns on, then once the cells become activated, another transcription factor, MYF5, turns on, and then, as I already told you, MyoD, the master muscle regulator, turns on. Then later MyoG or myogenin turns on, and then myosin light chain turns on. So it's a nice progression. So we know exactly where we are in the process of differentiation. And this is key because as you're learning about stem cells, how to harness them for regenerative medicine, you want to know, where is my stem cell? Which markers is it expressing? And we're going to come back to this when we think about using stem cells in disease models, because it's not every cell is not the same, right? And so when these markers are turned on in this spectrum, it tells us a lot about how these cells are going to be useful in clinical situations. And so my main point here is that really the molecular markers of the cells are well understood. And the next phase is really thinking about how to harness these, how to purify these cells, how to really take advantage of their properties for downstream applications for either understanding the basic biology of the muscle stem cell or for tools to isolate these for use in regenerative medicine. Now, don't worry about all of the complexity of this slide, but I just want to point out that we have a lot of examples of receptors that are expressed on these satellite cells. And you see in the center, you can see the beautiful transcription factors that are expressed in the satellite cells, the PAX7, PAX3, MIF5, MyoD, depending on the stage of how that cell is in its spectrum of differentiation. But what you may not be aware of is that there are a lot of other cell surface markers that are unique to these cells. That's important because we need to be able to purify these cells to be able to better study them, to use them in potential clinical applications. So once we have the ability to isolate these cells with some of these unique receptors, for example, we routinely use things like NCAM or VCAM, these extracellular surface markers, to purify these cells and then study them. Well, why do we want to study them? Well, as I told you, what we want to understand is where are these stem cells in any given time in culture, right? Because they might be at a quiescent state, they might be at an activated state. And this is really determined by different types of division that these cells are undergoing. So they could be undergoing a symmetric or an expansive phase where they're making more copies of themselves. Or they could be going in asymmetric division where they're keeping the original stem cell properties, but then they're functionally differentiating to become a more myoblast-like cell, which, as I told you, is going to eventually give rise to a fused myotube and a functional myofiber. So the location, the ability to purify these cells, to isolate them with these cell surface receptors, to study their ability to differentiate and divide are key properties and key things that we do as muscle stem cell biologists. So one thing you don't know yet, one thing I haven't told you, is that the stem cell itself, the satellite cell, and this is true in a number of different adult stem cell systems in particular, is that it's not just the property of the stem cell itself. It's not just the ability of the cell to symmetrically renew or to asymmetrically divide to give rise to a different muscle cell, the myoblast. It's the ability of these cells to receive cues or signals from what's called the niche. And the niche is different for every adult stem cell system, but the satellite cell niche is pretty well understood in the adult, in the sense that we know a lot about where the cell is located. And where a cell is located tells you a lot about how it's going to function in the niche. We also know a lot about the signals that are nearby, right? So we know that there are growth factors that are being secreted that tell this stem cell to maintain as a stem cell. Basic FGF is a great example, Wnt7a or other great cytokines that we know a lot about that really talk to the stem cell in that environment and say, hey, we need you to remain a stem cell. There are other signals that a stem cell might see, for example, damage signals that might tell that stem cell it's time to make more muscle. And so those cells, those self-renewal signals would go down as the differentiation cytokines or signaling factors or ECM factors would go up. So this is really exciting biology, is understanding the interplay between the stem cell and the niche. But that's at the individual, immediate satellite cell environment. And it's not just signaling within that local milieu. It's actually, it's not just signaling within the nearby the satellite cell, but it's also within the local milieu. And so essentially, as I told you, there are a lot of other cell types that are nearby that are going to talk to the satellite cell like neurons, like blood cells, like fibroblast cells, pericyte cells. All of these are examples of cells within that local environment that are going to play a role in really the dynamics of the stem cell itself. How many stem cells are there at any given time? How many stem cells are going to be activated in the muscle? What cues are being responded to upon injury or damage in the muscle? And finally, not just the local environment, but systemically, there are a lot of regulators, and so there's many different levels and or cells, signaling factors within the niche that regulate how the stem cell itself behaves, and so that tells you how important it is to really regulate stem cell behavior in any given organ system. So, for example, in the skeletal muscle, there are really known, well understood regulators, cytokines, and cell types, immune cells, for example, which you're going to hear more about in terms of, when there's damage in Duchenne muscular dystrophy, for example, these immune cells become activated, and these talk to the stem cell, right? So there's a lot of interesting biology in the niche that is key to understanding how to regulate the fate of that cell. Now, I like this cartoon because I think it really simplifies it for you, and it also shows you that there's a lot of really tight crosstalk that's going on during muscle regeneration. So the stem cell is the satellite cell, right? That's the key cell that's going to give rise to new muscle. But there are other cells nearby that's going to, regulate the biology of that cell. So shown here is really the, a nice close-up view of cells nearby: blood cells, pericyte cells, fibroblast cells. A lot of exciting research is now on FAP cells, these fibroadipogenic progenitors. That are really important in the context of regulating muscle regeneration in the context of muscle diseases. So let's think a little bit about the biology of regeneration, because that's really an important role of the stem cell, is that, upon injury or damage, there's an cascade of activation cues that occur that really tell the stem cells, hey, it's my turn now to get in there and make more muscle. And this occurs, it's pretty well studied in animal models, in that we understand the timing of when this regeneration occurs. And so essentially, after injury, there's a timing of 5 to 10 days in which the muscle is really becoming remodeled, and over time, after injury, eventually the muscle is completely regenerated. As you can see in the 30-day cross-section of muscle, that you have central nucleation, which tells you that those muscles have been regenerated. And so these are really hallmarks of how we understand the stages of regeneration, which is really, in the context of skeletal muscle, catalyzed by the endogenous muscle stem cell, the satellite cell. So because we know so much about the endogenous muscle stem cell, the satellite cell, we kind of have a goal as to how we want to achieve getting that cell, or thinking about using that cell in regenerative medicine. But one of the challenges is the inability to really obtain large numbers of that endogenous muscle stem cell, particularly in the context of thinking about giving these cells back to patients. So one of the sort of revolutionary paradigm-shifting discoveries in stem cell biology is the ability to generate what are called induced pluripotent stem cells. So I've been talking about embryonic versus adult, but within pluripotent stem cells, there are different types of these cells that have the ability to differentiate into all 3 germ layers. And these are either called embryonic, which means that they're derived from the inner cell mass of discarded blastocysts, or they're derived from reprogramming an adult cell or a somatic cell back to the pluripotent state. These are called induced. You also hear them called reprogrammed. Now, why would you want to think about embryonic or induced pluripotent stem cells for muscle diseases? Well, one thing is that, as I told you, we're learning more and more as a field to think about how to push their differentiation potential to make things like muscle satellite cell equivalent cells from these. And so how do we make these induced pluripotent stem cells? Well, this is really catalyzed by discovery of Shinya Yamanaka, where he overexpressed key transcription factors that are important to pluripotent stem cells, namely OCT4, KLF4, c-MYC, and SOX2. So these transcription factors essentially remodel the somatic cell to tell it to become a pluripotent stem cell. And so this process of reprogramming is now becoming more well characterized and is becoming more routine, such that we can do this from a lot of different cell sources, not just fibroblast cells, but other adult cells. And so this is really a unique pluripotent stem cell type. Now, pluripotent stem cells, as I said, have a really unique potential in contrast to adult, in that you can take a pluripotent stem cell and you can differentiate it into any cell type. Well, you can do this from a human cell, right? So you can take, in the case of induced pluripotent stem cells, you can take a human somatic cell, you can reprogram it back to a pluripotent stem cell, so-called human-induced pluripotent stem cell. Then you can differentiate it into a patient-specific pool of muscle, a patient-specific pool of liver cells, of cardiomyocytes, of neurons. And this is really exciting because you can study the basic developmental biology of human cells in this process of just differentiating these cells. So this is remarkable. Another thing is that because we have a patient-specific population of a patient-specific pool of these cells, then we can test things that we've never really been able to do before, like, for example, toxicity in the context of cardiomyocytes. So drugs that are going to be given to patients can now be tested on patients' own heart cells, right? And so this is remarkable because it really provides a unique preclinical tool that we can now do in the context of patient-specific genetic backgrounds and epigenetic factors. And then finally, what you've heard the most about and what is really exciting is pushing these cells for use in cell replacement therapy. So we can take cells that are differentiated from pluripotent stem cells and give them back to patients. And so this is really, I think, revolutionizing our ability to perform personalized medicine here using these cells. So how do you do that? Well, it really depends on the lineage of choice, but in general, stem cells in vitro have their own little in vitro niche, right? So they either grow on another layer of so-called stromal or supportive cells, or we provide them with certain cytokines that tell them to maintain their expansive properties to self-renew. So when we take these away, we take away this stromal, we take away these cytokines, they immediately think, okay, I'm not a pluripotent stem cell anymore. Now I'm going to immediately differentiate, and they start a cascade of differentiation. Now the challenge is, as I already told you, how to tightly control this to tell them you're just going to make oligodendrocytes. You're just going to make satellite cells. So this is where we are in the context of thinking about how to tightly control their trajectory of differentiation. So we can make three-dimensional spheres, and this is really unique because if you look at these spheres, you can actually section through them and see specific cell types within those spheres. You can later harness these cells and then think about giving these back, testing them in more functional assays, which is really what we're doing in the field, is to analyze their properties. How good is the cell that we make from a pluripotent stem cell to an adult stem cell equivalent? So these are important future directions in the field of pluripotent stem cell biology. Now, in the context of Duchenne muscular dystrophy, which is what we're all thinking about, how can we harness pluripotent stem cells for modeling the disease, for testing preclinical therapeutics, for understanding the development of muscle in the context of Duchenne muscular dystrophy, and for understanding cell-based replacement therapies. Well, what we do is simple, as I've sort of already alluded to. We obtain patient cells, and then we develop the iPSCs. And these are just an in vitro cultured pluripotent stem cell line that we grow routinely in a dish in the lab. Once we have these iPSCs, we spend a lot of time characterizing them for their pluripotency ability. They make tumors, they make benign teratomas, they have the ability to differentiate into all three germ layers, they remain karyotypically stable. Once we understand the properties that we've made a patient-specific cell line, for example, from a DMD patient, then we ask questions about how do these cells make muscle, and we probably compare them to a wild-type patient iPSC line. So how does the DMD line differ in culture from a wild-type or non-mutated iPSC line? And one of the things that we think about is how to make cells that we can give back to the patient. And so in the context of Duchenne muscular dystrophy, cells we think about are things like cardiomyocytes or skeletal muscle progenitors, which would be akin to the adult muscle stem cell, the satellite cell. Now, I know what you're thinking. Because we've obtained these from the patients, there's another concern that we have to think about, and we'll come back to that. But essentially, that is because these patients have mutations in dystrophin, which you've already heard about. And so we have to think about how to correct this as well as combine these with stem cell-based approaches. And we're doing that as a field moving forward on that. Once we've established a cell type of interest, for example, cardiomyocytes or skeletal muscle derived from DMD iPSCs, then we can use these as a tool to test drugs that might uniquely affect those specific patients, right? And so we can ask a lot about drug specificity, drug toxicity, off targets in the context of real patient muscle, which is really remarkable. And then finally, if we can understand the biology of how to get those cardiomyocytes or skeletal muscle cells, we can give them back to patients. And the beauty of human-induced pluripotent stem cells is that they should avoid immune rejection because they're derived from the patient cells themselves. But these are still sort of things that we're testing in the field. Now, I told you that we can use iPSCs to model, to potentially generate cells to give back to patients. But why would we do this? Why is this relevant in the context of DMD? Well, as I already told you, muscle has a beautiful endogenous stem cell. And this cell makes more muscle, and upon injury, this gives rise to new myofibers. However, in the context of patients that have Duchenne muscular dystrophy or DMD, they're undergoing, continuous cycles of injury, regeneration, making new muscle, that their muscles are really susceptible to injury. And this is a constant cycle where they're trying to activate their stem cells to make new muscle. Now, what you can see is really the process of how these satellite cells do that, which essentially is to, upon injury, activate cues that tell the cells to divide, to give rise not just to more stem cells, but to activated progenitors called myoblasts, which are going to align at the site of injury, fuse to make new muscle. And as I said, these activated satellite cells, undergo asymmetric division that generates a new daughter cell, the myoblast. And then these myoblasts make new muscle, but this is a continuous cycle. So eventually these stem cells become exhausted in DMD, which is why we think about repairing or replacing these cells in the context of stem cell replacement therapies. So because these cells, the endogenous muscle stem cells or the satellite cells, have a constant regeneration cycle, there's a heavy burden on these cells. There's constant muscle damage. And eventually, as is the case in most adult stem cell systems, these cells are eliminated. They're exhausted. So what happens is the stem cells lose the ability to make more copies of themselves. They also lose the ability over time to differentiate, right? Because there's just fewer and fewer of these stem cells available. And when you change the environment, the muscle environment or the niche, you change the properties of that stem cell. So as the stem cells decrease, other cells fill in the void, right? So these cells are things like fat cells, scar tissue, things that make fibrosis. This changes the muscle environment. This changes the stem cell properties. So this is something that is really at the forefront of understanding stem cell biology and Duchenne muscular dystrophy, and how loss of these cells really affects muscle biology over the lifetime of these patients in DMD. So I've told you a lot about the properties of stem cells, their really unique differences between adult and embryonic, but how can we really think about how stem cells are going to be important for DMD? Well, as I've sort of already alluded to, one thing we can think about, because these stem cells are exhausted over time, is to give stem cells back to the patients, right? So you can give patients back normal stem cells that can continuously self-renew, right? And this is a new tool really to regenerate muscle. You can also use induced pluripotent stem cells, right, which are derived from somatic cells, right, back to the pluripotent state and then differentiated from those cells. And those cells have the potential to avoid immune rejection. However, there's a challenge of the need to correct the patient's cells, right? So that's sort of a combination therapy that would be required in that context. Another important thing that you could think about, because I've told you a lot about stem cell biology regulation, is that there's a lot of cytokines in that local environment. And so some cells might be useful, not really for making new muscle, but for really regulating that environment. So producing things like helpful cytokines, proteins, ECM, matrix factors that either remodel the niche or talk to the stem cell itself and regulate the biology of that cell. So finally, the real kind of, next wave, I think, or direction of stem cell biology for DMD is thinking about personalized approaches, right? And so how can we harness the potential of the patient's own cells for either cell-based therapy or for basic aspects of developmental biology, and things like drug screening, as well as patient-specific strategies are really what we're thinking about for using stem cells for DMD. So these are all really exciting, important avenues that are really going to come to fruition, I think, for DMD in the next 5 to 10 years. And I think that we're all going to benefit from understanding the basic properties of these cells and harnessing them for Duchenne muscular dystrophy. So I think really the next phase will be trying to harness the use of these cells in personalized genetic medicine. And what I'm going to do in the next mini lecture is to really tell you about where we are clinically with using these cells. So what are the differences in, and where are we, either preclinically or clinically, about testing the properties of these cells, about using these cells for patients with Duchenne muscular dystrophy. And I think that really in the next 5 to 10 years, we're going to really find some important information, pros and cons about using the different stem cell populations, which you'll hear about in my next mini lecture. So stay tuned. Thank you.